Last class we all started with polysaccharides, isn't it? So there we saw the homopolysaccharides, various homopolysaccharides. What are they? Starch. They are of two kinds, right? The storage and the structural. In the storage kind, we saw starch and glycogen, and in the structural, we saw cellulose and chitin, isn't it? About starch, it consists of amylose and amylopectin structure. Amylose is unbranched. Right? What kind of linkage is there? Alpha 1,4 linkage between the monomeric units. And how is the branching in amylopectin? Alpha 1,6 linkage. So both are forming a double helical structure around one another to form the starch. Then glycogen is similar to amylopectin of starch. How is it different? It has got more extensive branching. Alright. Then we learnt about cellulose. Right. So what is it in the backbone of cellulose? Made up of glucose. Is it branched or unbranched? Unbranched. What kind of a linkage you see? Beta 1,4 linkages. Alright. And then next chitin is monomeric unit is N-acetyl glucosamine residues linked to one another by what kind of a linkage? Beta 1,4 linkage, right? So then we try to see what are heteropolysaccharides, and in heteropolysaccharides we saw various kinds of heteropolysaccharides. So there we discussed about glycosaminoglycans and peptidoglycans. So peptidoglycan is present in the cell wall of bacteria, and what is it composed of? NAM and NAD. Alright, alternating NAG and NAM, they form a straight chain. Alright, and several such straight chains are linked together by cross links made of tetrapeptide, alright, and pentaglycine for gram positive and pentapeptide for gram negative bacteria. So what are the carbohydrates which give virulence to the bacteria? O antigens and teichoic acid. So this teichoic acid is present at the gram positive bacteria attached to the peptidoglycan, and it confers virulence. O antigen, there are several unusual monosaccharides which are present on the surface of gram negative bacteria, and we saw various kinds of glycosaminoglycans. So what are they? Various different types of glycosaminoglycans. Hyaluronic acid, chondroitin sulfate, keratin sulfate, dermatin sulfate, and heparin. So which is that glycosaminoglycan which does not have uronic acid? Keratin sulfate. And that GAG which does not have sulfate group? Hyaluronic acid. Alright, that which is responsible for the pliability of skin? Dermatin sulfate. And which is more prominently present in the cartilage? Chondroitin sulfate. Okay, so that comes in presence of sulfate group either in the 4th carbon or on the 6th carbon. So accordingly it is chondroitin 4 sulfate or chondroitin 6 sulfate. Then that GAG which is more prominent in the hoofs, the horn, etc. is keratin sulfate, and heparin is got the property of anticoagulant in nature. So that which is the natural anticoagulant is? Very good, I am very pleased. Like these kind of questions I will not be asking in the test. Alright. So we just started with glycoconjugates, right? So I was So what are glycoconjugates? They are those polysaccharides which are either attached to proteins or lipids, alright, and they have several properties. Particularly, they are present on the cell membrane. They are integrated in the cell membrane. So if you have studied the fluid mosaic model, you might have studied that along with the lipid bilayer, there is the presence of glycoproteins and glycolipids. So that is how they are having a role there. So they mainly help in the interaction of the cell to another cell or the cell to its extracellular matrix. And why it has to interact? In order to carry out the various biological activities. Alright, so interaction with proteins and lipids, these polysaccharides are glycoconjugates. They also help in the movement of the cells and they have several biological roles. Alright, they help in several activities by interacting with the proteins or any kind of molecule which is present in the extracellular matrix. Alright, so there are several types of glycoconjugates. They are proteoglycans, glycoproteins and glycolipids. First, talking about proteoglycans. What are they? First and foremost, these proteoglycans are found in the extracellular matrix. They are macromolecules present on the extracellular matrix. And what are they composed of? They are composed of the glycosaminoglycans. Not any, but the sulfated glycosaminoglycans along with a transmembrane protein. This protein is called as the core protein. So it is covalently bound to this membrane protein called as the core protein, and you have the sulfated glycosaminoglycans attached to this core protein. So this forms the structure of peptidoglycan. Proteoglycan. So if you look at the structure, it consists of a basic core protein to which this glycosaminoglycan is attached, and the attachment is through a tetrasaccharide. Over here, the attachment of the glycosaminoglycan is through a tetrasaccharide or a 3-sugar complex. It is glucose, galactose, like that, galactose and xylulose. So like that, it has got 3 to 4 saccharides and a serine residue bound to one another. So through that kind of a linkage, it's a glycosidic bond between the protein and the carbohydrate. Alright, so at the core protein junction, the carbohydrate, the heteropolysaccharide is attached.
Ma'am, the last point again, ma'am.
See, the joining between the core protein and the heparan sulfate or any sulfated glycosaminoglycan is by its 3 polysaccharide residues. It's 3 residues and the serine residue of the protein. So that is the glycosidic linkage. Alright, galactose, xylulose, and you have serine. So this will be the joining bondage between the protein, the core protein, and the glycosaminoglycan. Alright, so if you look at the structure, how these proteoglycans occur in nature, they are generally present in 2 major families. Okay, one are called as the syndicants, and these syndicants, they have a transmembrane domain and an extracellular domain. So that means if this is the cell membrane, onto the cell membrane you have a domain which is the transmembrane domain through which the core protein is attached, and you have the extracellular domain which is extending outward, and in this extracellular domain you have several chains of heparan sulfate which has been attached. Alright, now similarly you have another class of proteoglycans. These are the glypicans. These glypicans are attached to the membrane through a lipid anchor. Alright, it's a lipid anchor through which it is attached. Alright, and it has got a globular protein structure on top. The core protein becomes globular outward on the extracellular portion, and here also it has got several heparan sulfate residues attached to itself. So generally what happens is that both these proteoglycans will be shed into the extracellular space. Alright, they are not going to be found completely attached throughout their life cycle, but when at the time of requirement for binding to certain proteins, they are going to be released out of the membrane surface. So who is going to release them are certain enzymes like protease enzymes or phospholipase enzymes. This lipid anchor, it is called GPI. It is glycophosphoinositol, if I'm not wrong. Okay, it's called the GPI, the lipid anchor, through which the glypicans attach to the surface of the cell membrane. Alright. So even though it is attached through a lipid anchor, so because it's a lipid anchor, you need a phospholipase in order to cleave the attachment of this glypicans from the membrane. The same way, even syndicants have to be cleaved from the membrane so that they are free moving in the extracellular matrix. So why they have to be cleaved? Why they have to come out from the membrane surface is because they will have to form a kind of binding with certain extracellular complexes like ligands or certain proteins. They are going to help in the binding and carry out several biological activities. Now you might ask, what are these biological activities? How it is going to help? So there, based on the ability I'll repeat for those of you who have not understood. The proteoglycans can exist in two major families, two major categories. One is the syndicants and the other is glypicans. So syndicants are those which are both are bound to the membrane surface. Alright, both of them are bound to the membrane surface, and syndicants are like a long thread. Their core protein is present as a long thread where some portion of the core protein is a transmembrane domain and some of it is the extracellular domain. And onto this extracellular domain of the protein, several heparan sulfate residues are attached like a long thread, like a ribbon. Several of these heparan sulfates will be attached. So that will form the syndicant structure. Now this can also be present as glypicans. Glypicans are also proteoglycans which are bound to the membrane surface such that it will be attached to the membrane by its lipid anchor, and it has got a globular protein portion, and to it also few, compared to syndicants, few of the heparan sulfate residues will be attached. So these are the two ways in which the proteoglycans are attached to the membrane, and these will separate out. When they separate out at the time of requirement of their interaction to the proteins or any ligand, any molecule they will act as receptors to bind certain protein molecules to carry out biological activities. About that, I'll be explaining now. So if you look at the heparan sulfate molecule, okay, based on the ability to binding to certain proteins, this heparan sulfate has got two domains. Alright, one is the NS domain, which is highly sulfated domain, and the other is the NA domain or N-acetyl domain. So you look at the heparan sulfate here, it has got two domains, NA domain and NS domain. Alright, it is at this S domain, also called as S domain or NS domain, which is highly sulfated. So that is why it stands for S. Alright, so that domain is where the interactions are carried out. The binding of several proteins take place at the S domain of this heparan sulfate glycosaminoglycan, which is attached to a core protein. So let us see how that happens. So if you look at this structure, are you all understanding, online students? Aditi, did you understand what syndicants are and glypicans are? Are you there? So here, if you look at how the heparan sulfate interacts with the various proteins, there are several ways in which these syndicants will interact with the various proteins. So if you see here, you have the heparan sulfate having the S domain. So there is one way in which it can interact is by inducing a conformational change in the protein which interacts with it. Alright. So at the time of inhibiting a clotting process to take place, this is antithrombin. antithrombin protein will bind to the S domain of the heparin sulfate and it will undergo conformational change. Only when antithrombin will undergo this conformational change, it will help in the binding of several important factors. Clotting factors will bind to, if you see here, factor XA is a clotting factor which will bind to the antithrombin, and that binding will bring about the prevention of clotting. Alright, so the S domain of heparin sulfate will interact with the antithrombin and bring about a conformational change in this antithrombin. So this is why it has to be released into the extracellular matrix. So that when it is released, at the time of some injury, the clotting has to be prevented. So at such circumstances, protease will come and lyse the syndicates. The heparin sulfate will be made available to the antithrombin, and this antithrombin, once it binds to the S domain, it will undergo a conformational change such that its sites available for binding of factor XA are easily available. So that binding will bring about the inhibition of clotting. So this is one way in which it can act. Now another way in which the S domain can help in the interaction is by bringing about two different proteins close to one another. So if you see here, it has got antithrombin and thrombin. Alright, both of them will bind to two S domains which are adjacent to each other. So because of that, they will come in close proximity to one another and they will bind to one another also. So S domain will help in bringing about two different proteins closer to one another, and that will help in their interactions. Alright, so because of that, again it will help in inhibiting the clotting process. So if you see, S domain is actually a helping axis. Alright, it is just going to help the two proteins. Alright, you want to carry out a particular activity. Fine, come, I'll bring you both together. I'll give you both an interaction so that that particular activity can be carried out. So it is just a mediator which will bring the two proteins closer to one another, or it will change the conformation. It can also act in several other ways. So how other ways? It can also act as a co-receptor. Right? For example, if you see the fibroblast growth factors, alright, the fibroblast growth factors always come as dimers. Alright, so these fibroblast growth factors will bind to the S domain, and the S domain will act as the co-receptor along with the fibroblast growth factor receptor. So in the presence of the S domain and the FGF receptor, the ligand molecules will also come and bind. Alright, so the binding of, particularly when there is low concentration of FGF, the binding of ligands, fibroblast growth factor ligands, are facilitated by the presence of S domain which will act as the co-receptor. So this is another way in which S domain can act. Another important way in which the S domain can help in the binding of extracellular proteins is that because it consists of heparin sulfate has got several negative charges on it, right? So it is electrostatic in nature. It has got several negative charges on its domains. So because of that, all the positive. Others, can you hear me? Aditi, Namrata, can you hear me? And can you see me? Can you hear me, online students? Urvi, have a look at your system. Reconnect or restart your system otherwise. Alright, so get back here. We were talking about another way in which it can interact is because of the presence of negative charges on its surface. It can bind the positively charged molecules like several lipoproteins. This is how the virus particles come and bind to the surface. On the cell surface, it comes and binds to the heparin sulfate. Alright, so how? There are various ways in which There are various ways in which the heparin sulfate moiety of an proteoglycan will help in the interaction of the extracellular proteins to their ligand receptor complex, to form the ligand receptor complex. So you can understand what are the biological roles of this proteoglycan, isn't it? Apart from being present as syndicants and glypicans, they are also present as aggregates. Proteoglycan aggregates are called as aggregants. Okay, these aggregants are nothing but Supramolecules, okay, like a brush, bottle brush, they are present. So that is the, what you see here, the orange thread in the center is the hyaluronan, to which several chondroitin sulfate and keratin sulfate will be attached. Even in this kind of a form, it is available in, it's present in nature. Alright, so mainly they help in the interactions carried out in the collagen and extracellular matrix, and they have the role of elasticity or tensile strength. So apart from interactions, the syndicants and glycans are involved in the interactions. The aggregants are involved in the giving the support system or the structural, they are acting as structural polymers. And how are they present? They are present like a bottle brush. The hyaluronan is the rod onto which these several brushes are, the threads of the brushes are present, and these threads are made up of chondroitin sulfate or keratin sulfate. Alright, so these are the various ways in which the proteoglycans can exist in nature. Alright, any doubts? Shall we proceed?
Ma'am, the main road is
Hyaluronan or hyaluronic acid. Alright, so this will be attached to the cell surface. Resilience means that without breaking, you can bend, you can expand it, you can fold it, and it will not get bent. Alright, basically the elasticity. Which portion do you want me to repeat? S domain. You want me to just repeat it? Alright, I'll go very fast, I'll brush up fast so that you will have a brief outlook on it. Alright, the S domain, see basically heparan sulfate consists of S domain and the A domain, Na domain and the S domain, and this S domain is where several proteins come and bind. Alright, so it can act in several ways. One is to change the conformation of the proteins. After the protein binds, like for example antithrombin, after it binds to the S domain, it will change in its conformation, which will help in the binding of another factor to it, and this will inhibit the clotting. Another way in which S domain can interact is that two different proteins will bind to the adjacent S domains, and that interaction also will favor inhibiting the blood clotting. S domain can also act as the cofactor by which the fibroblast growth factors come as dimers and bind to the S domain. This S domain as a cofactor will help in the binding of the ligands to their respective receptors. When the S domain, because consists of high negative charges on their in their structure, several positively charged lipoproteins can come and bind to this S domain. So in this way, we are asking.
yes, ma'am. Protein-protein interaction, ma'am. It has to be thrombin and antithrombin.
Thrombin and antithrombin is not necessarily, these are just the examples. There are several such biological roles of proteoglycans. Here I have just mentioned the 4 different ways in which it can interact. But there are several other examples also which you can find, where you will see how the S domain brings about interaction between 2 different proteins or proteins and any other factor for that matter. Alright. These are just 4 basic examples I have given here.
And then what is the function of the Na domain, ma'am?
Na domain is just the, it does not have specific role in the binding. It is just flanking the 2 S domains. Alright, otherwise you will have a stretch of S domains. Right, so you shouldn't be having a straight stretch of S domain. So because that will not help in the binding correctly. Like here 2 different proteins has to be interacting to one another. If only when it is flanked by an acetylated domain, it will help in the binding. And if you look at the structure of heparan sulfate again, you have got these sulfate domains, sulfated groups which are little farther from one another, right? So that is why you have S domain present a little one next to the other, and in between you have the acetylated. Alright, the acetylated domain is nothing but the carboxylic acid group in the structure. Understood? Fine, let's proceed. So we saw what aggregants are. Now coming to glycoproteins. So glycoproteins you must have already studied a lot while understanding the fluid mosaic model. Come inside. Fast. So late. So talking about glycoproteins. They are proteins which are covalently bonded to several carbohydrates. So that proteins, these glycoproteins over here, the proteins are said to be glycosylated. That means they are attached to the carbohydrate residue, and these are generally attached to the outer membrane. So how the glycoproteins are different from proteoglycans is that the proteoglycans get released into the extracellular matrix, right? But here the glycoprotein never gets released into the matrix. It is always bound tightly in the lipid bilayer, and to them also, onto the surface of the glycoproteins also, several proteins will interact. Alright, the same way as how the proteoglycans function, these also have got several interactive functions with the proteins present in the extracellular matrix. Alright. So these glycoproteins are not restricted to the extra cell membrane of only the entire outer cell, but it is also present inside the several organelles found attached to the membranes of several different organelles inside the cell, like lysozymes, the lysosomes, the Golgi complex, etc. So what is this glycosylation like? Very simple to understand. One is the N-linked glycosylation, in which the carbohydrate moiety of this glycoprotein is attached to the protein through the amino group. Alright, so that is N-linked. Where, for example, you have N-acetyl glucosamine residue attached to the asparagine residue of the protein through the amino group. So that becomes the N-linked glycoprotein. These N-linked glycoproteins are more frequent. You can see them more common. Similarly, you have the O-linked. O-linked, the N-acetyl galactosamine is attached to the serine residue through an oxygen linkage. So this is O-linked. So either the glycoproteins are present as N-linked or O-linked. The glycosylation is N-linked or O-linked. Alright, so there are several functions which the glycoproteins can carry out. Alright, extremely vast spectrum of functions are there for the glycoproteins. So you have enzymes, transport proteins, hormones. So generally every protein which is released out gets glycosylated, alright, in order to make it functional. So this glycosylation is a kind of post-translational modification, alright, by which every protein, once it is being synthesized and released, it gets glycosylated in order to become functional. For example, for the hormones. Alright, FSH, what is FSH? Follicle stimulating hormone, or thyroid. Any of the proteins released gets usually glycosylated. Alright, so what is the role of the carbohydrate which is present in this glycoprotein? Mainly, it helps in the folding of this polypeptide to make it biologically active. Alright, and also it prevents the proteins from undergoing any kind of a proteolytic damage. Then talking about glycolipids, very similar to glycoproteins in their function, in which the lipids are attached to the carbohydrate residues, and these are also found attached to the cell surface just as the proteins are attached. So in the case of proteoglycans, you have long chain of heparan sulfate, several such heparan sulfates. Like aggregans, you have large amount of these glycosaminoglycans which are attached. But here there is a protein which is in the core membrane, transmembrane, and you will have short oligosaccharide residues which are attached onto the surface. Or it will be a protein, it will be a lipid to which short chain oligosaccharides are attached. So that is how you can distinguish between the proteoglycans and the glycoproteins. The same way for lipids. Cell signaling. I suppose glycoproteins have got vast roles. It interacts with several ligands, several proteins on the matrix. Alright, so as far as my knowledge concerned, it has got a role in cell signaling as well. Alright, fine. Then coming to, you must have already studied gangliosides, right? Gangliosides, the other ma'am must have mentioned. So there again they have got lipids and carbohydrates interacting with one another. So that is a typical classic example of glycolipids. Alright, and they also have several roles. of bindings. Okay, so we talked about sialic acid. Gangliocytes contain sialic acid. What is the sialic acid? Exactly, N-acetyluramic acid. N-acetyluramic acid, how do you get that? It's an interaction between manosamine, okay, pyruvic acid, manosamine, N-acetyl manosamine and pyruvic acid forms N-acetyluramic acid, and that is nothing but sialic acid. So generally you have sialic acid present in the gangliocytes, which are also a glycolipid. Apart from that, certain gram-negative bacteria possess lipopolysaccharides on their surface. Alright, so some of these lipopolysaccharides are toxic in nature, right? So on the surface of E. coli, generally you see lipopolysaccharides in which you have several lipid residues, okay, arranged side by side, to which you have a small core protein and you have an O chain or the O antigen. It is conferring the antigenic property. So you have even certain roles of lipopolysaccharides in the pathogenic species of certain bacteria. Alright. So apart from that, we have to know what lectins are. Lectins are nothing but certain proteins, alright, which are synthesized in the in certain plants, particularly in plants, animals, bacteria. You see these lectins being synthesized. Mainly they have high affinity towards the carbohydrates. They are not always bound, but they always have high affinity to the carbohydrates. So here I have just shown you a few examples of lectins here. So there are certain plant source lectins, animal source lectins, viral and bacterial lectins. Right. So some of you answer these questions. Amylopectin has got glucose residues. The branching is at the interval of 24 to 30 glucose residues. Okay, which of the following is a heteroglycan? Heteroglycan. Chitin is a heteroglycan. It's a homopolymer, right? It is a homopolymer. Inulin. Have you heard of inulin? Inulin is a fructose. It's a repeating unit of fructose polymer. Alright. You generally see them in the roots. The storage roots of plants will contain this inulin. Agar, of course. Agar is a heteroglycan. It has got two or more different, like N-acetyl glucosamine residues. It forms several cross-linking to form the agar. Okay, then that glycosaminoglycan, which does not have uronic acid, is